Welcome back to The Explainer. So you're sitting in a clinic, a phlebotomist is drawing your blood, and you're probably thinking, it's just a routine plumbing job, right? Well, scratch that. Today we're diving into the hidden science of a blood draw. We're going to see how this everyday event is actually a high-stakes, chemically precise procedure where microscopic mistakes can completely alter your medical outcomes. Okay, let's dive right into this with a quote that pretty much sets the baseline for all medical testing. A perfectly performed test on the wrong patient's specimen is a dangerous laboratory error. I mean, really think about that for a second. You could literally have million-dollar lab equipment and the most advanced medical science in the world at your fingertips, but if the initial identification is botched, all of that technology means absolutely nothing. It's a terrifying thought, but it highlights exactly why there is so much strict science behind the collection process. To keep those errors from happening, here's our roadmap for this explainer. We're going to cover the 3 testing phases, proper patient and specimen ID, the difference between blood, plasma, and serum, the actual chemistry of those colored tubes you always see, the absolute crucial order of draw, and finally, why specimens get rejected.
All right, let's kick things off with Section 1, the 3 testing phases, or as I like to call it, the journey begins. So every single laboratory result relies on a multi-step journey, not just a quick moment inside a machine. First up, we've got the pre-analytical phase. This is the unsung hero of medicine, basically everything that happens before the actual testing. We're talking patient prep, the collection itself, labeling, and transport. Next is the analytical phase, which is the actual testing inside the lab. And finally, the post-analytical phase, like validation and reporting. But here's the kicker. The vast majority of potential errors, they happen right at the beginning, in that pre-analytical phase. That's where most human intervention happens, so that's where we need to focus first.
Moving on to Section 2, proper patient and specimen ID, getting it right from the very start. Listen, the stakes here are incredibly high. Relying on looks is a catastrophic failure. You can never, ever just go off a feeling that someone kind of looks like the patient on the form. No way. Institutional policy requires strict, objective identifiers. Before a needle ever even touches an arm, a patient has to be identified using things like their full name, their unique hospital or lab number, and their date of birth. And right alongside identifying the patient is identifying the specimen itself. A label with the patient's name, lab number, date, and time absolutely must be applied immediately, right at the time of collection. If someone finds a tube without a label, you never ever try to guess or just relabel it without following strict protocols. An unidentified specimen is a totally useless specimen because the risk of a dangerous mix-up is just way too high. Okay, Section 3, blood, plasma, and serum. Time to decode the fluids. Now, what we generally just call blood actually comes in distinct chemical states. If you take whole blood, which is your cells plus plasma, and put it in a tube that stops it from clotting, the liquid portion is called plasma. Now, plasma contains water, electrolytes, proteins, and this is crucial, clotting factors, including one called fibrinogen. But if you let the blood clot naturally and then remove that clot, the liquid left behind is called serum. The sharp contrast here is that plasma still has its fibrinogen, while serum basically doesn't, because that protein gets completely used up forming the physical clot. Depending on what the lab needs to test, they're going to specifically ask for whole blood, plasma, or serum. Which brings us to Section 4, the chemistry of colored tubes. They are way more than just pretty caps. Now, what's really interesting here is that every single cap color acts as a cheat sheet for the chemical environment inside that tube. Take the lavender or purple tube, for example. It's packed with EDTA, an additive that prevents coagulation by basically grabbing onto calcium ions. This perfectly preserves blood cell morphology for hematology tests. Then you've got the light blue tube with sodium citrate. That one also binds calcium, but it does it reversibly, making it the standard for coagulation tests. Moving down, the green tube uses heparin to enhance antithrombin, stopping clotting for plasma chemistry. And if a doctor needs to check your blood sugar, they reach for the gray tube. It holds sodium fluoride, which literally stops the cells from consuming glucose, locking in those blood sugar levels for testing. I actually want to pause for a second on that light blue tube, the one with sodium citrate used for coagulation tests. That crucial 9-to-1 ratio represents the exact parts of blood to sodium citrate required inside that specific tube. If a phlebotomist underfills it even slightly, this exact chemical balance gets thrown completely out of whack, and it ruins the coagulation result for the patient. It requires immense, absolute precision. All right, Section 5, the crucial order of draw, preventing chemical contamination. Because these tubes are filled with such powerful chemicals, there is an exact, highly specific sequence required during the draw.
Why? To avoid ruining the samples through invisible, microscopic cross-contamination. As the needle switches from one tube to the next, tiny amounts of the chemical additive can actually carry over. To prevent this, the standard CLSI-based sequence dictates drawing in this order: blood culture bottles first, then the light blue Citra tube, then the red or bold serum tubes, followed by the green heparin, the purple EDTA, and finally the gray fluoride tube. And this brilliantly illustrates exactly why that order matters. Imagine a phlebotomist draws the purple EDTA tube before a chemistry tube. That invisible EDTA carryover on the needle, it introduces excess potassium into the next tube. And because EDTA chelates calcium, it also binds up the calcium in that second tube. If you look at the patient's chart later, it'll show a fake, totally dangerous potassium spike and falsely decreased calcium and magnesium. A perfectly healthy patient could literally be treated for a heart-stopping potassium overdose, entirely because of the order the tubes were drawn in. Crazy, right? To avoid this kind of nightmare, medical professionals use memory aids. One highly satisfying mnemonic to help lock in that sequence is clean cats sleep happily every Friday. Culture, citrate, serum, heparin, EDTA, fluoride. It's a simple little sentence, but it literally saves lives by maintaining chemical purity. Finally, Section 6, Why Specimens Get Rejected, the pitfalls of the draw. So, the biggest physical threat to a beautifully drawn specimen is something called hemolysis. This is the destruction or rupture of red blood cells, which causes the release of their intracellular contents directly into the plasma or serum. When this happens, the specimen turns an alarming pink or red color. But what actually bursts these cells? Well, often it's just rough physical handling. A difficult venipuncture, using a needle that's too small with excessive pulling force, extreme temperatures, or even simply shaking a tube way too hard instead of gently inverting it. When those cells burst, they dump massive amounts of potassium and LDH right into the sample, causing entirely spurious, invalid lab results. Hemolysis is a huge culprit, for sure, but labs will also reject samples for other reasons. Maybe it was drawn into the wrong container, or there was an insufficient quantity, like we saw earlier with that strict 9:1 citrate ratio, or maybe a specimen that requires whole blood ends up clotted. Add to that leakages, unlabeled samples, or excessive delays in transport, and you really start to see the absolute minefield lab technicians navigate every day. And remember, these aren't just administrative inconveniences. They are major patient safety issues. So as we wrap up this explainer, I want to leave you with this question. Next time you're sitting in that clinic chair and you see those brightly colored tubes lining the tray, will you recognize the invisible chain of custody and the intense chemical precision that dictates your medical care? A blood draw isn't just a simple pinch. It is an absolute masterclass in chemistry, timing, and protocol. Thank you so much for joining me on this deep dive. Keep asking questions, and I'll catch you in the next explainer.